Now, Chapter 15, Section 3. How do you regulate the synthesis and the breakdown of glycogen? Well, glycogen breakdown is caused by an enzyme known as glycogen phosphorylase. Thing is, glycogen phosphorylase itself comes in two forms, A and B. A is catalytically active. B, much less. Okay, it is active still, but its kinetic rate is greatly reduced compared to A. B, as we see here, sits there. It's less active. It is liberating some glucose molecules from glycogen, but not a lot. Until outside signals, hormone, glucagon, or epinephrine, boom, hit the cell. This is going to tell phosphorylase B kinase, activate this, which is then going to phosphorylate two serine residues in phosphorylase B. Remember, addition of inorganic phosphates will cause a change in the overall shape of the enzyme, making it go from B to A. A form is the highly active. A goes and cuts, releasing glucose 1-phosphates from the glycogen strands, the glycogen polymer. And it'll keep doing it. Go, 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 go. Until A is reverted back to the B form by the enzyme PP1. Glucagon epinephrine activates phosphorylase B. B converts to A. Cut. Something activates PP1. A is dephosphorylated and reverts back to B. There's this whole series of enzymatic steps, signal pathways that will activate, convert B to A. Just got done talking about the fact glucagon, epinephrine can push this conversion to B to A, meaning we're going to get lots of G1Ps flooding the cytoplasm. The mutase will convert G1P to G6P, which can then go into glycolysis or have the phosphate group cut off and the glucose transported out to the bloodstream. You've seen this before in previous courses. I know I taught this in Intro Cell and Molecular. Epinephrine binds its receptor, activating a G protein. One of the targets of the G protein is activating adenylyl cyclase. Adenylyl cyclase takes ATP, turns it into cyclic AMP. Cyclic AMP will activate pKa. Active pKa activates phosphorylase B kinase. Remember, it's a kinase, so it's going to strip off a phosphate from ATP, targeting something. Well, what is it targeting? It's activating, remember, B, phosphorylating B to turn it into the active form A. So glycogen phosphorylase A goes, cuts glycogen, releasing G1P. G1P will then be converted to G6P to enter into glycolysis, energy. Boom. Glucagon does relatively the same thing. G protein, cyclic AMP, boom, 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 boom. Glucose into the bloodstream. All is good. This is the muscle cell. This is the liver cell. Notice, though, one glucagon, one epinephrine, one G protein activated. One G protein activates adenylyl cyclase. Adenylyl cyclase makes about, each one will make about 20 cyclic AMPs. 20 cyclic AMPs go and activate about 10-ish each pKa. pKa goes and activates, those 10 go and activate 100 phosphorylase B kinase. Phosphorylase B kinase will convert B to A, slightly active to the active form of about 1,000 molecules. Those 1,000 will then go and cut about 10,000 G1Ps, which will now be utilized in the cell, released to the bloodstream. And that's for each time an epinephrine or a glucagon binds its receptor. That's a lot. This is amplification. 1, 20, 100, 1,000, 10,000. We also see that calcium is a regulatory mechanism. Just think about it for muscle contractions. Nerve impulse down, central, hops peripheral, down to the neuroskeletal junction, neuromuscle junction. We see the depolarization and then the triggering of the release of calcium from the sarcoplasmic reticulum, which causes the myosin and everything to muscle contraction. Well, to keep doing that muscle contraction, you're going to need energy. Going to need more liberated glucose from the muscle glycogen stores. All right. Turns out calcium can bind directly to and activate phosphorylase B kinase. So whether or not you have epinephrine or glucagon hitting in the muscle cell, that release, the flooding of the cytoplasm with calcium, which is going to trigger a muscle contraction, is also going to trigger the activation of phosphorylase B kinase, which means you'll get the shift from phosphorylase B form to the A form and cut, releasing sugars, glucose, G1Ps, for use by the muscle cell to generate more ATP. If your muscle cell is burning through ATPs, means you're going to have lots of ADPs, lots of AMP. So you had epinephrine hit, and you had a lot of cyclic AMPs made, and that cyclic ring was cut. So your AMP levels are high, and your ATP levels are low. This also can activate phosphorylases, which will then trigger G1P's release. Lots of things enter into the glycosidic citric acid cycle. Oxidative phosphorylation, ATP levels go up, AMP levels drop. If the cell is happy with its ATP, it's already got high levels of ATP, well, these two don't matter. This high levels of ATP allosterically inhibits phosphorylase, blocking these two. Now that PP1 talked about a few slides ago, that takes A and converts it back to the less active B form. PP1 cleaves off the phosphates from the two serine residues, which allows A to shift back to the B shape. That's it. That's what is it occurring. Now, contrary to what a lot of students want to think, they think all of B gets turned into A, all A gets turned into B, and it's a one or the other complete. And no, glucagon's being released, epinephrine's being released. You get varying levels of phosphorylase B kinase. So at any given moment, some of the B's, if not the majority of the B's, are turning into A's. Still going to be some B's there. PP1 goes and starts converting back almost immediately A's to B's. Not all, just some. If you have glucagon and epinephrine still hitting there, signal, well, guess what? You're still going to have the shift where the majority of the B's going to A, fewer A's shifting back to reverting back to B. So you get this balance thing going. More A, less B, more B, less A, more A, less B, depending upon who's getting the signal, who's getting the greater push. Now to help, once there's enough glucose has been liberated from glycogen. So you get that reversion from A back to B to help PP1 along. We have come to find out that phosphorylase A has 2 allosteric sites. 2 allosteric sites that are the correct shape for glucose. Remember all those glucoses, those G1Ps that are being released from glycogen. If that level hits high enough, hits above a certain threshold, well, you get these interacting, which is going to cause this phosphorylase A to have a greater affinity for PP1, greater chance of that reversion from A back to B to occur. As long as there's glucose there, boom, B is going to be predominant. Now, once the glucose levels drop, then we can see more of that conversion of B to A, less of the reversion of A back to B. So it's a shift. It's a constantly shifting thing, but is it shifting one way or the other? What are the cues that the cell is receiving? Now, that's for the breakdown. What about the building up? Well, synthase. Synthase is an enzyme, ends in A, so what does it do? It synthesizes. What is it synthesizing? Glycogen. Well, it turns out glycogen synthase also comes in 2 forms. A, active, B, inactive, just like we saw with the phosphorylase. But it's switched. A is unphosphorylated. B here with the glycogen synthase is a phosphorylated inactive form. This is the opposite of what we saw with the phosphorylase AB. What's phosphorylating? Well, glycogen synthase kinase. Ends in A's, it's a kinase. What's it adding the phosphate group to? Glycogen synthase, also known as GSK3. GSK3 turns A into the phosphorylated B inactive form. How's this working? Well, inactive form. You don't need glycogen right now. Got enough. Glycogen synthase, the enzyme that makes the glycogen, is phosphorylated and inactive. PP1 is going to pull off, pluck those phosphates and turn it into the active A form. Here's the thing. What activates PP1? Insulin. Remember, insulin is the hormonal signal that tells your liver, pull all the sugar it can out of the bloodstream, convert it into glycogen, store it away. The counter to the insulin is the glucagon epinephrine, which we just talked about 2 slides or 3 slides ago, which will trigger phosphorylases to break down glycogen, release sugars into the bloodstream. So these 2 are counterproductive of each other. They counter each other. This says store it, this says liberate it. So if you have glucagon, high levels of glucagon epinephrine, PP1 is not going to become active, which means you're going to stick in a B inactive form, and all that glycogen is going to be liberated. Glucose by glucose, I should say. If you have lots of insulin and little to no glucagon epinephrine, well, store. A form says store it. Store it. When levels get high enough, GSK3 is going to convert A to the phosphorylated B form, the inactive form. PP1 will convert it back. GSK3 turns it here, PP1. Now, if insulin is saying PP1 do its job, insulin is telling GSK3 stop, don't do your job. Epinephrine inhibits PP1, but it also activates GSK3 or allows GSK3 to do its job. But before GSK3 can do its job, CK2 has to come in and lay down at least one phosphorylation. So that's what they're showing here. CK2 phosphorylates glycogen synthase, which then allows GSK3 to come in and further phosphorylate GK synthase A phosphorylate. I should say phosphoglycogen synthase A. Now, why is that? Well, it turns out GSK3 can't just go and start doing its job. It has to have a phosphate, a serine already phosphorylated, so that it knows where to position itself. You look here. Here is the glycogen synthase primary structure. Okay? Sequence of amino acids. GSK3 has two sites. One site is the active site. The other site is a priming site, or in my mind, a positioning site. GSK2, as we see right here, GSK2 laid down this phosphate group right there. It laid it down, and it acts as a positioning tool for GSK3. This phosphate enters into this priming site. It positions the active site on the next serine to be phosphorylated. This ghost image here is showing that after this phosphorylation event is done, GSK3 will shift. The priming site will now be on the new phosphorylated serine. And hey, look, the next serine is in the correct place. And then the next, in the next, in the next. For all of this to occur, what we have come to find out is all these proteins will collect together, glycogen targeting proteins. Will act as a scaffold, so everything is here together. If one of them is active, the other is inactive. They're right there next to each other. We see this balance occurring. Phosphorylase active, synthase inactive. Synthase active, phosphorylase inactive. Who's being phosphorylated, who's not? They all work together in tandem. Build up, break down, build up, break down, build up, break down. Now, as I've mentioned, and I'm not going to beat this dead horse, in the liver, insulin and glucagon are in a battle. Insulin activates glycogen synthase. How does it do that? Inactivates GSK3. Keeps synthase in the active A form. Activates PP1 so it can take more of the inactive B, dephosphorylate it, and turn it into A. Flip side of that coin, glucagon inhibits PP1, aids in GSK3 activation. Keeps this going. Keeps the breakdown, the liberation going. When you look at the regulation of this, up, down, insulin-sensitive protein kinases will yield increased levels of active PP1. High levels of active PP1 means low levels of phosphorylase kinase. Low levels of phosphorylase kinase, decreased levels of glycogen breakdown. High levels of PP1 means more glycogen synthase is being activated, B to the A form, which yields glycogen synthesis. High levels of insulin, high levels of protein kinase B being activated, which means GSK3 levels will be lowered. Low levels of active GSK3 means high levels of glycogen synthase. more glycogen being synthesized. So you can sit there and go through this over and over. High levels of insulin. Hexokinase 2 takes lots of the G1P or glucose. And converts to glucoses, which will then be sent into glycolysis. Takes G1P or takes just glucose, turns it into G6P, straight into glycolysis. Lots of glycolysis occurring. Flip side of it, which is why the orientation has changed. This was for high blood glucose. This is for low blood glucose levels. Glucagons released. Increased levels of cyclic AMP. Increased levels of active pKa. Phosphorylase kinase active. Glycogen phosphorylase. Breakdown of glycogen. Releasing G1P. High levels of pKa means glycogen synthase levels are going to drop. Shift from A to B because GSK3 was going to be activated. Synthesis decreases. High levels of pKa, we're going to see decreased levels, decreased levels, decreased levels, decreased levels of all the enzymes of glycolysis. Because you want to take the sugars and you want to be able to release them into the bloodstream because you got low blood sugars. You don't want to have the sugars entering into glycolysis to be turned into pyruvate and ultimately released as carbon dioxide and lots of ATPs. So you're going to see this shift. High, low blood sugar levels. Now, a lot of what we've been talking about is dependent upon whether or not you're talking liver, you're talking muscle. Glucagon acts on the liver. Glucagon has absolutely nothing to do with muscle. Epinephrine works on both. Now, ultimately, muscle, glycogen, liberates G1P, G1P converts to G6P, G6P enters in glycolysis, you get pyruvate, citric acid cycle, oxidative phosphorylation, ATP levels shoot up. Liver, well, it depends. Was it the glucagon or was it epinephrine? If it's glucagon, G6P will be converted to just glucose, which will then be dumped into the bloodstream. Pyruvate, thanks to gluconeogenesis, will be converted back to G6P, turned into glucose, out into the bloodstream. If it's epinephrine hitting, glycogen, glucose 6-phosphate, pyruvate, high levels of citric acid, glycolysis, citric acid cycle, oxidative phosphorylation, lots of ATPs. So it depends which cell and then which signal is triggering the event. What we know now is carbohydrate and lipid metabolism are integrated, are hormonal regulated. What turns on one turns off the other. Well, I don't want to say turn on, turn off. Let's say shifts. Shifts from one, shifts to the other metabolically. There are allosteric mechanisms that if the cell has lots of carbohydrates that are available, it's going to shift metabolism from fatty acid to carbohydrate. Carbohydrate centric. Still some fatty acid occurring, but it's mostly carbohydrate. If carbohydrates are going down, but there's lots of fatty acids, well then it's going to shift. More fatty acid, less carbohydrate. And then back and forth. Never the same, never completely off. Just kind of shifting. They're still occurring. It's just who's at the forefront, who's in the background.